Hey everybody, today we are going to be diving deep into the wonderful world of bacteria and how it has become immune and resistant to many of our antibiotics today. While some of these bacteria can be good and beneficial for us, others only tend to attack and harm our bodies.
The bacteria itself releases enzymes that digest tissues and attacks cells that are around it. Because of how fast the bacteria can duplicate itself, the same process is continued throughout the body at a significant rate, getting rid of our needed cells left and right.
But here's where the greatness of science comes in. We have ways of stopping these attacks. Scientists have figured out how to both slow down and kill the bacteria. This is thanks to antibiotics. There are a couple ways that these antibiotics are able to fend off these bacteria. One of the most common approaches affects cell wall synthesis.
This method is able to prevent the enzymes responsible for building the bacterial cell wall from being produced. Now, once the cell wall is destroyed by the antibiotic and is unable to be reproduced, the bacteria dies.
However, the more the bacteria is in contact with these antibiotics, the more resistant they can become. One way is for the bacteria to go through a process called transformation.
This basically means that the bacterium takes in DNA that has a resistance for an antibiotic and makes it a part of itself. This resistant bacteria can then duplicate itself with a new string of DNA, making it almost impossible for the antibiotic to work the right way.
In order to avoid a world where the bacteria is completely resistant to all of our antibiotics, doctors like to keep a list of DOLRs, also known as drugs of last resort, to use in case a patient has this specific mutated bacteria in them.
We keep the use of these drugs to a minimum because bacteria can't be resistant if they have never had contact with these certain antibiotics before.